right, everybody, welcome to the Chapter 2 lecture. Well, we'll see how far we get through this in about 15 minutes or so. But Chapter 2 is basically just about molecules of the cell. This is all stuff that everyone has seen before in previous biology classes, and it is considered to be review for microbiology. So we're going to zip through this pretty fast. So this is review stuff. So molecules of the cell, we're going to be dealing with lots of them. So this is a refresher. But basically, and last time I talked about how there are organic molecules in the cell and there are inorganic molecules of the cell. You already know the difference, that there is carbon and hydrogen in organic molecules and not both carbon and hydrogen in inorganic molecules. So inorganic molecules that are in the cell, well, stuff like water. Before you're like, wait a minute, I bought a bottle of organic water at some food co-op somewhere. Like, nope. Water is an inorganic molecule. If you see a bottle of water from Fiji Island or something and it says that it's organic, you go to the manager and tell them that's a bunch of BS, because water is by definition inorganic. It has hydrogen, but it doesn't have carbon, and it is an inorganic molecule. So water is the medium through which everything in the cell is basically hanging out in. Water is the basis for the cellular environment as well. So there's a ton of water in the cell. Also, other molecules like, well, O2 is also an inorganic molecule. Prevalent in the cell. We breathe it in, we breathe out CO2. CO2 is really a waste product for us and for most microbial cells as well, but it's there, it's inorganic, even if we're getting rid of it. Also things like phosphate ions. It's usually a -3 charge to that. This is a phosphate ion. Very common in the cell. Calcium +2 ions, magnesium ions, stuff like that. Of all the ions that hang out in the cell, I'd say the phosphate group is probably the most important. Before we get into organic molecules, I do want to just say that because water is the universal solvent and it's a major portion of the cell, depending on what is dissolved in the water, you're going to have a difference in pH. pH stands for potential hydrogen. It's a measure of how acidic or basic something is. So I just want to quickly go over to our PowerPoint. And sorry, I can't seem to open this up in PowerPoint, so we're just looking at the pictures here. Try to ignore the little open button down at the bottom. But the pH scale runs from 0 down at the bottom where the red is, where it's very acidic, on up to 7, which is pH neutral, and on up to 14, which is very, very basic or alkaline. Most alkaline things are not things that we ingest or that any cell considers to be food. Things like bleach or sodium hydroxide, drain cleaner, lye, household ammonia, milk of magnesia, detergents. Cleaners tend to be very basic because that stuff destroys cells. And in fact, bleach is going to be around pH 12, very basic, and that's what you're using to kill bacteria in your lab setup. pH neutral is 7. Anything above pH 7 is basic, even slightly basic, like blood here is listed at around, I don't know, I'm guessing that is like 7.25. That would be slightly basic. Things that are slightly acidic would be like milk. That would be a pH of 6.7 or something like that. We don't usually think of milk as being very acidic, but it's slightly acidic. It has some of the lactose gets converted to lactic acid. Urine tends to be a little acidic. It'll be anywhere between 6 and 7. Unpolluted rainwater. But look at this: black coffee, beer, vinegar, cola, lemon juice. A lot of things that we eat and drink, citrus fruits, for example, are going to be acidic. We tend to be okay with eating acidic things, but even things that are just slightly basic, slightly alkaline, tend to taste really kind of kind of gross to us. Like if you eat baking soda is basic. It doesn't taste very good. A lot of proteins that we eat in meats would be around a neutral pH. So that's the pH spiel. I am not going to ask you on an exam what the pH of household ammonia is. I just want you to be aware of the pH scale, and also that pH neutral, a pH of around 7.0 is neutral, and most cellular enzymes and other proteins function best at a neutral pH. So most cells, including your own, and a lot of bacterial cells, strive to maintain a neutral pH. Because if things get too acidic, that's going to mess with the shape of the cellular proteins, and it's not going to function well. If things get too basic, well, it can destroy, the phospholipid bilayer of the cell. So cells don't like that. Be also aware that a lot of cellular waste products are acidic, and so excreting them, will acidify the medium that a bacterium is growing in. It'll acidify the environment. If things get too acidic, not good for the cell either. So pH neutral is what most cells are trying to maintain as, the best optimal pH for them. All right, so getting back to organic molecules of the cell. You have some small organic molecules, and the bomb there would be adenosine triphosphate, ATP. ATP is energy currency of the cell, right? I learned all about that in a prior class. Please make sure you know how the cell uses ATP for energy. But because this is adenosine triphosphate, I might not know what this adenosine looks like offhand, but I know that means 3, and phosphate, it means it's got 3 of these phosphate groups on them. So let's take a look at what that looks like. Here we go, adenosine triphosphate. So these 3 phosphate groups, okay, they've got the PO4 right there, and they usually circle it in yellow like that because elemental phosphorus has a yellow color apparently. So oftentimes, instead of writing out that whole PO4 thing, what a lot of books will do is just put a P with a circle around it like that and say, okay, these are just 1, 2, 3 phosphate groups like that, instead of drawing all of this, because who wants to draw this hot mess? Nobody wants to draw this hot mess. Okay, so over here, actually, let me see if I can take a screenshot of this. Yeah, I'll bring this up so I can mark on it. Yeah, we'll use this blue. So we've got our 1, 2, 3 phosphate groups there. This part over here is the adenosine part. There is an adenine, which is this double ring structure here, and a ribose, which you should know by the ose, is a sugar. It's a 5-carbon sugar, and it exists in a ring structure. Put these two together, you get an adenosine molecule. So adenosine, 1, 2, 3 triphosphate right there. Energy for the cell. It is organic because it's got both carbon and hydrogens. FYI, there's a carbon right here at that corner there. If we don't put a C there, we know that there's a carbon here, there's a carbon here, carbon here, carbon here. Ooh, carbon there. Unless you see another letter like N, it's like, well, there's no carbon there, there's a nitrogen there. But if there's no letter at a corner, you know that there's a carbon there. So up here, there is a carbon there because there's not another letter to take its place. So that's just an abbreviation for those stick drawings. Just thought I'd mention that. Okay, I do not want to save that screenshot. There we go. So the ATP molecule is used in the following way, just as a quick refresher. And you know what? I'm not going to draw that whole thing. I'm just going to write ATP right there and put my 1, 2, 3 phosphates there. And this is actually the initial phosphate, the one that's hanging off on the end. So they're numbered this way, 1, 2, 3, like that. The way that it is used is that for energy, when the cell wants to do something that requires energy, it breaks off this initial phosphate group. So that you have this initial phosphate group, well, hanging out by itself, basically. Usually, or oftentimes it gets attached to another molecule. Like that, and that actually releases energy. So we say this bond here is a high energy phosphate bond. It releases energy that the cell can use to do stuff. There we go. Notice this says a DP now because this is not adenosine triphosphate anymore. They're only 1, 2, so it's diphosphate, adenosine diphosphate. So there's energy stored in that high energy phosphate bond right in there, and it gets released when that bond is broken. FYI, that the cell does not see this ADP molecule and think, oh, well, that's just a waste product. It's like, wow, wouldn't it be cool to get this molecule back together with its initial phosphate? Then, if we went backwards, we'd have ATP all over again. But that doesn't release energy. Actually, energy needs to go into that to make that happen. And that comes from basically the food that we eat. It comes from the food that we feed microbes. Microbes need food to make energy, and that energy is used to put this phosphate group back onto ADP to make ATP. And that will be the subject of, I think it's chapter 6. All right, so that's the small organic molecule of the cell, the most important one. There are large organic molecules, or let's just call them big organic molecules. I'm just abbreviating org for organic, molex for molecules. Stuff like carbs, proteins, nucleic acids, your DNA and your RNA. And oh, what's that last one? Lipids. Again, this is still all review stuff, people, so we're going to be buzzing through all of them. But for all of these, you need to know the following. Their functions in the cell. What is the function of nucleic acid in the cell? What are the functions of proteins? What are the functions of carbs? Why do you need to give carbs to cells? Why do your cells need carbs? Their function, basic structure, including how the monomers, or sometimes they're called subunits, are joined to form the polymer. Okay, a polymer is a molecule that is made up of many parts, and those little individual parts are the monomers or subunits. Sometimes they're even called building blocks. But poly means many, right? A polymer is a molecule that's made up of many smaller molecules. Okay, so we need to know that. Let's see, functions, their basic structure. Yeah, and their, let's say their basic chemical formula. Basic chemical formulas. Alright, so we are going to start with carbs, or short for carbohydrates, because they are my favorite. I love my carbs. I want to eat all the carbs. I love carbs. They are so tasty. Carbohydrates are common food source. Okay, sugars are carbohydrates, right? There are complex carbohydrates and simple sugars, but they're all still carbohydrates. Common food source. Sometimes we think of them as quick energy, not long-term storage of energy. That would be for lipids, but quick energy, common food source, carbs. But more than that, sugars, which are carbohydrates, are also a part of DNA molecules, part of RNA molecules, and I would add it's also part of the adenosine triphosphate molecule. You saw that ribose that was back there. Look at that right there, ribose. I'm not going to circle that because I don't want to have to take another screenshot, because we already saw where the word ribose is there. It's this ring structure down here with these two OH groups hanging off the bottom. That is a sugar. So they're a part of other molecules in the cell that are really important. They're also part of the bacterial cell wall. Peptidoglycan has a sugar that's a part of it. All right, and I have here, sugars can be used as a reserve food source in bacteria. Actually, our cells too will store some carbs long term. Again, we think of fats as being long-term energy storage, don't we? Lipids. But glycogen is a long-term energy source, and that's a carb. And some bacteria will do the same. Alrighty, so that's its functions. We need to talk about its basic structure. Okay, or basic chemical formula in this case. Carbohydrates all contain carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. What that means is that if we have a carbon with a carb with six, with 6 carbons, it's going to have 12 hydrogens and 6 oxygens. If I do C5, that means it's going to be H10, twice as many hydrogens as carbon, same number of oxygens. So, we could do this all day, right? C4H8O4. FYI, this one here is glucose. This one here is ribose. I do not know the name of this one here, but it's going to be something ose. Sugars end in ose, and I just want to make sure that you are familiar with that. Fructose is the same as ribose. It's just a different arrangement of atoms. Lactose, galactose. Sugars end in ose. Ose means sugar. There's so much vocabulary in science, isn't there? And it's really nice when scientists try to simplify things. So they're like, you know what? It's like a bunch of people got together and were like, you know what? Let's just name sugars with an ose at the end so that we know they're sugars, because we have enough other things to occupy our brains. We don't want to have to be memorizing 40 different things if we can just memorize one word ending right there. So they all have carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. Okay, they will also, so that's 1, 2, they will have OH groups, or what's called an alcohol group. Sometimes I call it an OH group. Sometimes it's called a hydroxyl group. It depends on its usage. Half the time I'm just going to say OH. Carbs have lots of OH groups. Thirdly, they're going to have something that looks like this. A carbon doubly bonded to an oxygen, but you know that carbon has to make 4 total covalent bonds. That's what those lines stand for right there. And usually there's a hydrogen at the end of it there. This is called an aldehyde group. If it's just this by itself, if we're only talking about this part, this is called a ketone group. And you'll see later on something that looks like this. This is a carboxyl or a carboxy group. We'll see that later too. Okay, let's get back to the PowerPoint here. Yep, they all contain an alcohol group. Usually have an aldehyde group or a ketone group, or at least they start off that way when they're simple sugars. All right, oop, oop, oop. Let's just build one, shall we? So let's build one. Let's say, okay, C5H10O5. If I'm going to draw this, oh, it's so easy to draw them linearly. Carbon loves to bond to itself, so I'll just string all 5 together like that. I know I got to have one of these things here, so I'll do that there. And the rest are going to make single bonds. And let's see, I got to do the alcohol groups and use up 4 more oxygens, so we'll go like that. And we'll fill in the rest with hydrogens, and I'll count them up and make sure I didn't screw this up. Okay, so we've got 1, 2, 3, 4, 5 hydrogens, 5 carbons, got those. Let's count up oxygens next. 1, 2, 3, 4, 5. Oh, got those. We're good. And the rest are hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yay, we got it. And I'm not seeing anything funky here. Every carbon is making a total of 4 covalent bonds. Like even here, this is 1, 2, 3, 4. The double bond counts as 2. So something like that. All right, but can you think of any other ways to maybe put these atoms together and have it still be C5H10O5? If you want to do it on your own, go ahead and pause this video and draw it out. But for those of you who are like, huh? I'm telling you that it is possible to arrange all these atoms and have it still be C5H10O5. Like, maybe I'll even still string them together in one line, 1, 2, 3, 4, 5 carbons, but maybe I'll put the doubly bonded oxygen there. And I'll put the alcohol groups hanging off this way. The rest will be hydrogens. Maybe something like that. Okay, let's see. So I've got 5 carbons. Don't need to count those up. We're good. Oxygens. 1, 2, 3, 4, 5. Got those. We're good there. Hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10. Yeah, I counted that one, counted that one. We're good. Hopefully I've convinced you that these are both, that this over here is C5H10O5 and this one is C5H10O5. They are, but they're not the same molecule. They've got a different arrangement of atoms, don't they? These are called structural isomers. Structural isomers are elements that have the same chemical formula, C5H10O5, but a different arrangement of atoms. Same chemical formula, but a different arrangement of atoms. One of these would be ribose, one of these would be fructose. Fructose and ribose I mentioned back here are both C5H10O5, but they're not the same molecule. All right, now I'm kind of stretching things here with drawing C5H10O5 linearly like this. because something funky happens. When you get up to 5 carbons or more, these things don't like to be in a long line. They get bendy, basically. And what happens, let's get another color. What happens is that this thing will bend over and this carbon will come over here and hook up with that oxygen, and this hydrogen will move to the oxygen. I cannot draw this because I'm a terrible artist. I'm a terrible animator here. But it forms a ring. So any sugar with 5 or more carbons forms a ring structure. Let me show you a picture of that. Right here. Yeah, monosaccharides have, we'll get to that in just a sec. So here we go. I'll take a screenshot here. And let me mark this up now. All right, so see how they've numbered the carbons over here? This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Look over here. Carbon 1 is over here. It has bonded to... Wait. No, that's the oxygen. See, I'm screwing this up. It short circuits my brain somehow. We have carbon number 4 here. Comes over and binds to the oxygen here and forms a ring. And then carbon 5 is going to be hanging off over here. Ribose exists in a ring form. You are not going to need to draw this for me. You are not going to need to tell me which carbon binds to what. You just need to know that they form a ring structure. So ribose is a 5-carbon sugar in a ring structure because if it's 5 to 8 carbons, it's going to form a ring. If it's over 8 carbons, it's actually going to be a different thing that we'll talk about in just a minute here. Note the difference between ribose and deoxyribose. The only difference is right here. Down here at the bottom in red, where I'm pointing for you. Here. Deoxyribose is ribose that has been deoxified. It's missing an OH there. Missing that. It's just an H. Subtle difference, but it's one of the differences between DNA and RNA. So there we go. Alrighty. Nope, this is a screenshot. Got to delete that. One more thing before we go on to saccharides. All right, another form of these exists. You can have, it's the same arrangement of atoms, but they can be in mirror images. So here we've got glucose. Here, do this in red. All right, we've got glucose here. It can exist in a mirror image, but you can't really argue that they're structural isomers. They're not. The arrangement of atoms is the same, just like the arrangement of fingers and thumb is the same in both of your hands, but they're mirror images of one another. These are called stereoisomers. They have the same arrangement of atoms, but are mirror images. They're mirror images of each other, and they are denoted by D and L. It just refers to which of the planes that it is. And I want to say that the ones that occur most in nature are actually the L versions of sugars. They're going to be L versions, not D. But they do exist, and they can be formulated in the laboratory, but they end up having different chemical properties. So which stereoisomer we're talking about actually does make a difference. Alrighty. Okay, so any simple sugar with 5 or more carbons forms a ring. Okay, any carb any carb with less than 9 carbons is a simple sugar, or what we call a monosaccharide. A monosaccharide. Saccharide refers to sweetness. Saccharide, there was a sweetener that was very popular back in the 70s. Well, you can actually get it now. It's in the pink packets. Sweet and Low is actually saccharin, which is just really disgustingly sweet stuff. Super sweet artificial sweetener. They called it saccharin. If a person is saccharin, like, oh, so-and-so is just so saccharin. I don't know if anybody even uses that word much, but it just means they're just ridiculously sweet. Like, I don't know, think of someone who's just over the top sweet. It's like, oh my gosh, they're so saccharin. And almost falsely so. That's what that word means. But monosaccharides, the word saccharide refers to carbs, refers to sugars, to carbohydrates. Okay, but we can put together 2 carbohydrates. Like here we've got one monosaccharide, and here's a second monosaccharide. We can put them together to form a disaccharide. I can't spell saccharide today. And then if we add 3 or more monos, equals what we call a polysaccharide. The way they are put together, there we go. How are they joined? Okay, disaccharides. Oh, lactose and sucrose. Yes, sucrose is actually table sugar. It's a disaccharide. How are they joined to each other or polymerized, we say? How are they made into a polymer? This way. Through what's called a dehydration reaction. The hydrogen of one, oops, I do not want to copy that, on one molecule gets up close and personal to an alcohol group on another, and you pull a water molecule out of there, and it's called a dehydration reaction. Let me draw that out for you a little bit better. Okay, let's say, and I'm going to erase all this stuff here. Give us some room. Give us a little room here. How do we make a disaccharide? Okay. All right. Over here. This oxygen here is kind of an electron hog, and it kind of pulls the electrons from these bonds a little closer to it. And that makes this hydrogen here a little loosey-goosey. And an enzyme that catalyzes this reaction senses this and is like, the hydrogen, this loosey-goosey hydrogen on this one simple sugar, I could get it up close and personal to the OH group on this other sugar. Pull them out together, and you know what 2 hydrogens and an oxygen makes. It pulls out a water. Pulls a water molecule out of there. So I'm going to move these here. Right. Oops, I erased too much. Oh, wait. Let me use this here. Oh, no, I pulled that out just right. Pulled out the water molecule. Now these 2 are left to bond to each other. Okay, it's not longer. I just can't move these whole structures closer together. But now you're going to have a chain that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbons long. That is a disaccharide. And it's created by a dehydration reaction. Dehydration reaction puts those puts those two together. And can you see that I could add a third one on there to like this one right here? Or maybe this OH group would attach to somebody else's, so something else. That would make a, not a trisaccharide, but a polysaccharide. That's what we call them. All right. Now these can be joined by, rings can join as well. So you could have glucose here and a glucose there, here a glucose, there a glucose, and join them together by a dehydration reaction between two rings. So that's what these little beads are for here. They're showing you all these circles here. Those are ring sugars that are joined to each other. Polysaccharides can be 3 or more. Usually they are really long. Like up here is cellulose. It's very lengthy. It's held together by, it's given a little extra structure by some hydrogen bonds. Glycogen, which is the one that I was saying is long-term storage for a carbohydrate. It's a little more branched. Dextran, a little less branched there. But basically things can branch out anyway, in any direction. And you can get some very complex carbohydrates that way. There we go. So let's see, we did mono and di and polysaccharides, ring structures, carbon-hydrogen to oxygen ratio, alcohol groups, aldehyde groups. We can move on next to nucleic acids. Nucleic acids are my second favorite. I love to play with DNA and RNA. All right, DNA, you already know, stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Let's talk about what their functions happens to be in a cell. And I'll come back here. They're called nucleic acids because DNA is discovered in the nucleus. That's where it hangs out. Okay, and we say that DNA is basically the genetic material of the cell. But what does that mean? It's the heritable protein. It's the heritable molecule. DNA is the genetic material of the cell in that DNA contains genes. And if you go look up the term gene, like in your ebook, in the glossary, it'll probably say something like the molecule of inheritance. But that doesn't give us the full story of what a gene is. Well, what do you mean? So, okay, we inherit our genes from our parents, fine. But what does that mean? What do they do? They just say you're going to be, 5'7" tall or something like that. They're going to say your eye color is going to be like this. They're going to say your ears and eyebrows are shaped in a certain way. Well, yeah, but the way they do that is through proteins. Another definition of a gene, let's expand our definition of a gene and say that it's an area of DNA that contains information telling the cell how to build a certain protein. So an area of DNA with instructions, let's say, for building a protein. So for every protein that's in your cell, there's a gene in your cell that has it. If your cell can make a protein, if your cell is able to make a particular protein, it's because it has a gene telling it how to make that protein. And this is kind of how it works. Let's say you want to build a house. Chances are you're not going to be like, okay, I bought some land, I have some supplies here, I'm going to build a house and I'll just wing it. I'll dig a hole and I'll bang a couple of boards together with nails and I'll put a bathroom wherever I feel like it, and I'll put a sink over here and the shower over there, and I'll put a window here. Whatever. No, you need a plan, right? And people can go online and buy what's called blueprints for their house. And there are even, wiki places where you can go and alter someone else's house plans and get all these house plans for free. So you need the house plans in order to build the actual house. Here we go, there's your house. All right, but chances are you're not going to just go straight from the plans to your house. It's probably not happening. I mean, are you going to do all the plumbing by yourself? Are you going to dig the hole yourself? Are you going to pour the basement cement by yourself? Do all the electrical work? No, you're probably going to hire a bunch of people to help you. You're going to hire an electrician to do all the wiring. You're going to hire a plumber to do the plumbing. You're going to hire a roofer to do the roof. Not too many people can do that all by themselves. So you need help, right? You need subcontractors who will help you build the house based on what your plans say to do. But the deal is, what if you only had one copy of your house plans? If you only had one copy of your house plans, you'd be like, okay, well, first I'll give the house plans to the people who were digging the hole for the basement. Okay, now you're done, give them back to me, and I'll give them to the people who are building the floor and the walls. Okay, can you give them back to me, and I'll give them to the plumber so that the plumbing people know where to put all the pipes. And then when the plumbing people are done with it, and you hope they didn't lose your plans or mess them up, spill coffee on them, so that you could then give them to the electrician so that they know where to put all of your plugs. No, you need copies of your plans. So you need copies of plans for your subcontractors. So the analogy, meaning that your DNA, those are your house plans. Those are your plans for how to build a protein. So these are protein plans or instructions. RNA, ribonucleic acid, these are the copies of the plans. Copies of the plans for how to build a protein. And then RNA gets out into the rest of the cell, gets together with all the ribosomes and everything, and then build a protein which is built based on what the gene and the copy of the gene said to do. And just like your subcontractors don't take your house plans, fold them into a shape of a house, and then give you your house as your little origami house and say, here, go ahead and live in that. They don't rearrange the pieces of paper that the plans are written on, and poof, it's a house. A house is made of different stuff than the plans. Likewise, proteins are made of different stuff. Proteins are made of amino acids. Probably heard that. I know you've heard this term before. This is review. So they are made of amino acids, whereas DNA and RNA are not made of amino acids. They're made of nucleotides. They're made of nucleotides. And nucleotides are not the same thing as amino acids. So the cell doesn't take the RNA made of nucleotides and fold it into a different shape and say, poof, it's a protein. No, it actually reads what the RNA says. It reads it. And in a later chapter, we will get into detail about how a copy of the DNA is made, and that copy is called RNA. And then how the cell looks at the RNA and builds a protein based on what the RNA says. It reads the RNA, like reading a book. And it uses the information in that book, or the copy of the book, really, to build a protein based on that. Okay, so that is enough for today. So I'm going to leave it there so that nobody gets overwhelmed with having to watch too many videos. But I will pick up from this a little bit later, and we will complete and look at what a nucleotide is made up of. And we'll cruise through proteins, and we'll get through lipids as well next time. So that's enough for the rest of this week, and I hope I'll see you in class on either Wednesday or Thursday, depending on which section you're in. So be well and have a good one.